So, as Maria was saying, I'm Enzo Ferrante. I'm from Argentina originally, but basically it's also my alma mater. I did my PhD here many years ago, or a few years ago at least. So I'm really happy to be back now as a visiting professor under the DATAIA program, so I'm very thankful to them. Today I will be talking about one of the research lines that has been more active in my lab during the last years, and I will tell you kind of a story from where we started to where we are right now, around this idea of anatomical segmentation, right? We will be talking about medical images, segmenting organs in medical images. That is the task in which we are focusing here. And how can we do that with deep learning models? And how can we also, at the second half of the talk, we will be discussing something that is really important for me, that is how to ensure fairness in these models. Fairness in the sense that these models are going to be fair with respect to different subpopulations. You know that AI today is taking over. We have a lot of models doing a lot of things, but we also know that these models may be biased with respect to certain subpopulations. So how can we take care of that in the context of image segmentation? I'll be commenting a little bit on some stuff we are doing in this field. So this is my team in Argentina, and the works I'll be presenting have been mostly done in collaboration with some of my students and colleagues there who are involved there. But let me start talking about one of the fundamental words I will be using today, that is that of anatomical plausibility. What do I mean by anatomical plausibility? Well, you know, here Maria, me, Sergio, and maybe some of you work a lot with medical images. We want to segment the structures to inform doctors about specific size, shape, texture, something. You want to extract information from these images. But in order to do that, we need to know where the organs are. So, for example, in brain images, you can see there some segmentations related to different tissues in the brain, or here some segmentations related to different organs in a CT scan. So these are organs that are always supposed to be there in our body, and they follow regular patterns. If we think about a brain, well, a brain looks like a brain, like it has a certain topology, and it cannot, for example, have certain like holes in the middle of the brain. Or the same happens with the liver, for example, right? We expect certain anatomy, certain shape, certain topology on these objects. So we will say that the segmentation mask is anatomically plausible when it lives in such space of plausible shapes, okay? And this is what we will be trying to encourage in our segmentation networks during this talk. How can we constrain the space of solutions so that we get things that make sense in terms of anatomy? When one thinks about image segmentation, in general, think about these kind of problems, right, in images in the wild. And here, you usually have a lot of data in this scenario. We have big data sets in computer vision with annotations like these ones, but we have low regularity in the sense that if you look at an image of a cat, well, the cat can be in completely different point of view. It can be rotated, like the shape of it when you are looking at it is not really regular. However, when you think about medical images, like the ones we will be talking about today, here, usually our data sets are much smaller, okay? We have really, like, not a lot of images, but we have much more regularity. Why? Well, because usually these data sets are taken in specific points of view. The organs, as I was telling you, follow a certain shape, and basically I want to take advantage of that to impose certain constraints in my model so that the segmentations that I get at the end of the day make sense. And most of the things I'll be talking here apply to the context of anatomical segmentation, like in this case, tissues, brain, wherever, but not to lesions. This is another really important area in which we work. When we want to segment lesions, things that are not supposed to be there in the body, like a tumor, like a white matter hyperintensity here, but you want to sometimes quantify. What I'll be talking about is not going to really apply to these cases. It's mostly going to apply to cases of anatomy. This is just kind of a disclaimer at the beginning of the talk. And then we can see why these ideas will not apply. This is the type of thing we want to avoid. If I have an X-ray image and I want to segment the lungs, well, these things are clearly not anatomically plausible. This thing there, neither. If I'm segmenting the heart in one of these images, there is a hole in the middle of the heart, that's not anatomically plausible. Same in this other case. So, or for example, in 3D segmentation, you are segmenting here white matter, and you have these holes here. This is not topologically correct. Okay? Why does it happen in general when we use deep learning models to perform these tasks? Why we end up having these non-anatomically plausible shapes? Where you know that in general the models we use here are CNNs, or well, nowadays more transformers, but when we started working on that, it was mostly CNNs, and CNNs have this property of local support, right? When I have the prediction for a certain pixel in the image, the area of the input image that affects that output is what we call the receptive field of this last neuron at the end of the day, and it's local. And that's a property of the convolution, basically. It means that the model is going to be looking at a certain area, but maybe not at the whole image, and so it's formed by a local patch somehow. So imagine I'm segmenting the heart here, but well, I'm segmenting the heart in this area, and this area is all white, basically, and it could also be something here in the background, right? So that's why sometimes we get these kind of problems here. CNNs are translation invariant, so as I'm telling you, for the network maybe it's the same thing if it's here or there, so it's difficult for the network to decide which label to put. And more importantly, and something we'll be talking a lot in this first part of the talk, the loss function that we use to train segmentation models is usually defined at the pixel level. So that means that this loss function will only be looking at a particular pixel, and I don't care where the error is happening. The way I will be backpropagating this error is exactly the same. If the error happens here, there, or there, because for those that are not maybe really familiar with segmentation, the way we define the loss function when we train up this network is at the pixel level. We compare and we just then average across all the errors in the different pixels. But I don't care about where it happened. So this property makes it really difficult to introduce global shape constraints, in the same that I want my segmentation to follow a certain shape, to live in a certain space of shapes. So how can we take advantage of the fact that we are dealing with a highly regular problem, in the sense that we are segmenting these structures that are organs, to encourage anatomical plausibility, sorry, via these kind of global shape constraints? And this is something we started to work a few years ago when I was doing my postdoc at Imperial with Ozan, who was the first author of this paper. And we proposed this simple idea. We want to learn embeddings which actually are going to contain information, global information, about the shapes that we are trying to segment. And we will use this embedding to complement the standard pixel level loss function, so that you have kind of a local loss function and also a global term that will talk about the anatomy, about the global shape. There are some kids there. So how can we do? Well, there are many ways in which we can learn embeddings from segmentation masks, but the easy one that we use here was to use autoencoders. So I guess most of you are familiar with autoencoders, right? A particular type of neural architecture where we force our model to reconstruct the input going through some low-dimensional bottleneck here. So this low-dimensional bottleneck will retain information that is global, that is useful to reconstruct, in this case, the segmentation mask. So we usually train these autoencoders following the noising autoencoder strategy. The idea is I will add some noise to the input, and I will train the model here. These are just fully connected or convolutional layers, wherever you want. Here in the middle, this has to be a fully connected layer for our model, so that it will retain information about the whole input. And so we input noisy segmentations, and we train the model to be noisy. This will help us to learn embeddings here that are useful, that are basically describing or learning information about the whole structure. So usually you do that, you get your noisy image, you go through what we call an autoencoder. So it's basically an encoder-decoder architecture. You take H is going to be the embedding, basically the output of your encoder there, and you will train it to minimize some loss function. In this case, for example, the mean square error of the reconstruction. Okay, so you get your image, the noisy one. You pass it through the encoder. You then pass it through the decoder, and you expect that whatever comes out of there is going to be similar to the input, non-noisy, the clean version of that. Okay? Adding noise helps us to learn some kind of more smooth embeddings that have a better support at the end of the day. So this is the way we will train an autoencoder, and this is the way we will get these simple embeddings here. Now, this can be trained only using segmentation masks, right? I have the segmentation masks of the organs I want to segment, and I can train one of these autoencoders. And as I was telling you, usually now if I train one segmentation network, a network to perform image segmentation, I will have this kind of pixel level measure that can be cross entropy, dice, whatever function you can use that we usually use for segmentation problems. But I will also, and this is the idea that we propose here, add a second term in the loss function. You will have this distance between the embeddings. What embeddings? The embedding of what my network produced and the embedding of my ground truth. So these embeddings contain global information. If I compare them, I will be really comparing the global structure of the segmentation, not anymore at the pixel level, but at the global level. And so this was the idea we proposed in this paper. We basically have our input image that we want to segment with a given network. In this case, this is the heart, and we'll segment the left ventricle of the heart. And so first thing you will do is you will pre-train an autoencoder with the segmentation mask. Okay? This will be done easily. Then we start training our network. You have your image, you get your. And the loss function will have two terms. The first term is going to be the standard pixel level, cross entropy, dice, whatever thing. The second term will be the Euclidean distance between the embeddings of what my network produced and the ground truth of my segmentation. This will make this comparison at these two different levels, and we try to encourage the network to the output, to be anatomically close. And this is the kind of things that you get. This, for example, is the segmentation you would get if you just work with the pixel level losses. This is your ground truth. And this is what you will get when you impose these kinds of global shape constraints. Okay? It will really take into account the global structure and not only the pixel and kind of local structure. Well, in terms of numbers, this also helps a lot. I'm not going to get here into the details. But then, at some point, we say, yeah, this is a nice idea. It helps us to improve anatomical plausibility in segmentation. Can we extend it to other problems in computer vision, particularly in medical imaging? For example, to image registration. Image registration is this idea of aligning images, right? I have, for example, two images of the brain, and I want to align them. But sometimes, what happens here is that to align the images, we use elastic transformations. We use non-deformable transformations. And sometimes, these non-deformable transformations are a bit aggressive. Okay? They kind of destroy the structure. So you use, in general, regularization methods, etc., to avoid these kind of problems when we are registering the images. But in this paper, what we proposed, it was to basically extend this idea of anatomically constrained neural networks for segmentation to registration. And now, you will have your standard registration network. This is a network that is created to register images. Basically, you want to register to align this image with this image. So you input both images as two different channels to your network. You input the two images as two different channels to your network. You produce a deformation field. And this deformation field that basically has one small arrow for every pixel indicating how the pixel has to move to be deformed so that the images end up being aligned. I will use it to deform one of the images and then measure this similarity metric to see, hey, how similar are the two images now that one of them was deformed? Usually you do that only using image information. So you train that to minimize some kind of similarity measure based on the input image information. and you use some regularization terms to avoid extreme faultiness here in this definition. However, what we proposed in this paper was to kind of use the same idea of these embeddings. Now, you will not only have your images, but you will also have segmentation masks with the structures you want to preserve when you are doing this deformation. And what you will do is basically apply the same deformation field now also to the segmentations and use the same idea of using this pre-trained autoencoder, computing the embeddings, and taking the embeddings to complement the pixel level loss. Okay? But this is for a completely different problem. It's for image registration. And why does it work? Why this idea of having complementary loss functions work? Well, this is something, one experiment we did in this paper that was really simple, but kind of tell us why these two are complementary loss functions. If you look here, I have a reference segmentation. And here, I created 4 different versions of this segmentation mask. In all of them, I swapped 120 pixels. So I basically moved from foreground to background, 120 pixels. So the number of erroneous pixels, basically, in all of them is the same. But here, I eroded the segmentation, so I keep it anatomically plausible. Here, I destroy the segmentation by removing the pixels in areas where creating these non-anatomically plausible shapes. And now, if I take the loss function between this and this, or between this and this, or between this and this, and I take the pixel level loss function, this pixel level loss function is going to be always the same. Why? Well, because I'm just changing 120 pixels, so there are 120 errors here. If I'm measuring the errors at the pixel level, this is what is going to happen. All of these errors are going to be the same. And this is the metric we usually use to train segmentation networks. Now, if you do that with our embeddings, we take the distance in this new space that we learn, well, what is going to happen is that these two are much closer than these two. So, if the network has to decide, I'm going to put 120 pixels wrong, well, it's going to prefer to do this than doing that. That's why these two loss functions are complementary to each other, and they help to obtain these more anatomically plausible segmentations at the end of the day. Okay, that was nice. Autoencoders learn the representation. We use this representation to regularize our learning process, and we end up having more anatomically plausible shapes, but imposing these constraints in a soft way during training. Now, for people coming from computer vision from the old days, you may know that we were really fan of post-processing steps. So sometimes we were doing some initial segmentation, and then we were kind of improving this segmentation to make it a bit better. So we said, can we use some of these ideas to basically improve arbitrary segmentations, improve the anatomical plausibility of arbitrary segmentation masks? And this was a paper we published a few years ago also on this topic, where the idea is extremely simple, but it worked really well in practice. And it was, let's say I have an image, and I have an arbitrary segmentation for this image. This one is clearly non-anatomically plausible, right? It has a lot of noise, it's horrible. We obtained that with a random forest model. So now that I have this, I want to do something to bring it back to the anatomically plausible space. And what can I do to bring it back to the anatomically plausible space? Well, I can take advantage of these autoencoders that are really good at doing that. So what we will do is basically train an autoencoder to bring non-anatomically plausible shapes to anatomically plausible space. What happens in general, you know that when we train autoencoders, there is this manifold assumption idea that all the segmentations that make sense, that are anatomically plausible, will live somehow in a low-dimensional manifold, right? Here in this line. When I have a corrupted segmentation mask, a mask that has a hole or that is non-anatomically plausible, usually what will happen is that this will be out of this manifold. And if I train an autoencoder and I take this non-anatomically plausible version of the image and I project it to the latent space and then I reconstruct it, what I'm going to be doing is basically bringing it back to the space of anatomically plausible solid. So this was the idea, and this worked pretty well in practice. So you pre-train one of these autoencoders using a really smart way of how putting this noise. It's not so obvious, not just random noise that I use when I train this. I use like geometric noise, different type of noise there. But at the end of the day, the idea is I have my images here. This is some initial segmentation that someone has provided to me. Here is the ground truth in the last row, where I want to really, okay, it doesn't work anymore. Here in the last row is the segmentation mask that I want to have, the ground truth. This is what you would get in the second row if you post-process using a standard method from computer vision, like conditional random fields, for example, that impose homogeneity to the segmentation, but not really these kind of prior shapes. And this is what you will get if you are coming from here, just using our post-DAE method, and only using label information, not image information. This is the interesting thing also about it. You come from label to label, not including any type of image, and you can see how you recover these anatomically plausible shapes. I think this ran out of battery. Sorry. You can measure that in different ways and show that this is improving. And actually here I have a video that should work to show that this is actually not cherry picking. Where you can basically see different cases of images, initial segmentation, what you get with our method, and the ground truth. So you can see that the method is really improving from these horrible things to this, which is of course not always perfect, but it's improving somehow. Okay, and it's just really cheap to use this post-processing segmentation. This was mostly work done by Agostina, one of my PhD students who just graduated. Well, then we say, yeah, this idea of denoising segmentations can be really useful, and this is a paper I'm not going to really talk about that. It's published here in PAMI, but it's just a paper that shows how we can use these ideas to improve segmentation quality during training as well. So here, the idea is that you will have images with segmentation labels and images without labels. And you want to leverage this image, these unannotated images. Okay? So this is kind of a weakly supervised plan. And what will be the idea? Well, the idea will be that for images that where you have segmentation labels, you will simply train your model as you have been doing always with the standard loss function. For those that you don't have segmentation labels, what you will do is you will segment them with your network, and then you will denoise this segmentation that you obtain and use that as supervision. Believe it or not, it helps a lot. And you can do that. I mean, you can use this denoise, it's called supervision by denoising, to take advantage of images that were not annotated. This was all for segmentation masks that are created at the pixel level, right? My segmentations were basically pixel masks. Now, in medical imaging, there are other ways in which we also do segmentation that is basically based on landmarks. When you have, like in this case, for example, landmarks that define your segmentation contour. Okay, so here I have an input image. I have this graph that is going to represent my segmentation. And in this graph, what I have, so you see that the difference, right, between going with the pixel level segmentation mask and a graph-based mask. So these graph-based masks are usually having all the graphs that represent my contours are going to have the same number of nodes. Okay, that means that for all the subjects in my data set, all of them are going to have correspondences. I know that the node 21 will be always positioned in this part. The node 0 will be always there. So if you have that, the idea is that we will construct a graph where V is going to be the set of nodes, in this case, simply the nodes that define the contour. A is going to be my agency matrix, okay, which is also going to be the same for all the subjects, because it's basically just indicating whether two nodes are connected or not. And I'm going to have a function defined on the graph that is going to be a function x that assigns basically coordinates to every one of nodes, or to every one of these nodes. So the idea is now that given an image, I don't want to generate the segmentation mask directly, but I want to generate one of these graphs. I'm going from images to graph. I'm changing the structure of what I'm predicting now. So how can we do that with neural networks? Well, what we proposed, because we are really fan of these autoencoders, was to use them, but so we will first kind of pre-train one of these autoencoder on images. Then we will pre-train an autoencoder on graphs. You know I can autoencode graphs as well and learn representations of graphs. And this you can do in different ways. We did it using graph convolutions, but you could use like fully connected networks, whatever. And the idea is that we will connect these two latent space. So I will take the encoder of the image part, I will take the decoder of the graph part, and I will connect them through a common latent space. And I will continue training this model with a loss function defined here at the node level. It's going to be basically the difference between the position where these nodes should be and the positions that were predicted by the model. And so this is interesting because it changes the structure of the output, right? I'm going from images now to graphs, not like before I was going to dense segmentation mask. And we tried different ideas for this decoder. If you perform PCA on all these shapes, you can just decode the V values of the PCA there, or you can use a standard fully connected variational autoencoder. This is also something I didn't mention, but these here are going to be variational autoencoders now, not the noising autoencoder standards. You know, variational autoencoders here will not encode like a deterministic value for these embeddings, but will encode the distribution of values. And then I can sample from there and basically generate shapes that are anatomically plausible, because this was trained with anatomically plausible shapes. So the only thing that this model will know how to decode are anatomically plausible shapes. And I'm kind of imposing now, by construction on my model, this restriction to say, hey, these shapes should be anatomically plausible. We use also graph spectral convolutions to implement the decoder that are basically convolutions based on the graph Laplacian that are really, really useful here. And these were the one that gave the best. We used a particular implementation of that based on the Chebyshev spectral graph convolution. And this is the architecture you end up having. You have the standard kind of convolutional encoder, and then for decoding, you have these graph convolutions that are going to have, that are somehow, this is somehow generative models model on graphs. Sorry. Then, like an extension, I'm not going to get into the details here, but you know that when we train segmentation models, there is something that makes a lot of difference in these kind of unit architectures that are the skip connections. These connections that go from encoder to decoder directly. So basically, we break this bottleneck that we usually have in autoencoders by putting skip connections here, which help in different ways. Help with the gradient when you are training so that it doesn't vanish. It also helps to recover details from the finer here, from the finer details into these layers here, avoiding this bottleneck. Otherwise, you end up always having really like a kind of smooth segmentations, not really detailed. And when you are using units to segment images and go to a dense pixel level segmentation mask, well, there it's really obvious how you just will basically take feature maps from here and take them to the corresponding level in your decode. Now, when you are working with images and graphs, it's not that obvious how to do that. How do I do this skip connection from images to graphs? So I'm not going to get into the details there, but we basically defined intermediate predictions for the node positions, and we were using these node positions to sample the feature maps in the corresponding space. Again, all the details are there in the paper. This is the journal version of that. And well, again, it improves a lot in terms of dice, Halfdorf, all the standard metrics we use to improve this. So if you compare with pixel level segmentation models, this is what you are going to get with our method, when this is a ground truth in the first column. And you are now avoiding, because now what you do is you take your segmentation and you just fill it when you want to create the segmentation level mask. And what you will see is that basically you are avoiding these kind of problems that are non-anatomically plausible. Why? Because our model is imposing like a strong prior by construction there in how this graph has to look like. So this also makes the model really robust to noise. Imagine, so this is another experiment we did. We took the images there and we generated occlusions, artificial occlusions. And so if you segment this with a standard unit, with a standard unit or any unit, these models that are pixel-based, you will get these horrible artifacts because the model basically doesn't know what to do there. It's like a black part. It didn't see it during training. Now, if you use one of our models, since it has this kind of strong prior on the shape it should have at the end, you will get these kind of segmentations that make actually a lot of sense. And actually here there is an experiment where we increased the size of these occlusion blocks, and we were seeing how our models, basically here in blue and orange, are not deteriorated, but the pixel level models deteriorate a lot as I increase the size of these occlusion blocks, what makes total sense. Also, if you have real artifacts, like for example here you have peacemakers, well, a segmentation model based on pixels will shall remove it. Our model, what you are looking is for anatomy, well, our model will be better there because it will leave it as part of the segmentation. And then now, since we are here going from images to graphs, well, this graph can be a 2D contour, but it can also be a 3D mesh. So we are now extending this method, and this is something we just submitted for evaluation, to volume to surface meshes. So now instead of going from 2D images to contours, we go from 3D images to graphs that represent the surface. And basically the same kind of architecture can be used here. It's just that you will be predicting a 3D graph at the end of the day. Okay. So just to give some, I mean, this is, yeah, at least I can skip maybe to go to the next part. So we really like how this model was working. And so we say, okay, why not taking all the big data sets out there with X-ray images? I don't know if you are aware, but there are many, many big data sets out there with X-ray images that people are using now for training models, etc. But these X-ray data sets do not have segmentation. So what we wanted to do, it was to construct a huge data set where we fused all the images out there, like in many data sets. So we have about 600,000 images here, with the corresponding segmentation maps that we produced with our model, which was really robust with respect to the different sites or whatever. So we did that. This is the data set. And what we did, it was to also not only provide the segmentation maps, but also provide an estimation of the quality of this segmentation. Of course, here we don't have manual annotations that we can compare because these data sets, exactly, they don't provide these manual annotations. But what we did, it was to segment them and generate this quality control metric that I'm going to explain you now how, like in a couple of slides, how to do. The code for that is also available. But I want to move now a little bit into the other problem I was commenting on bias on this kind of models. So, because it was a concern that actually was really important for us in this case when we were constructing this segmentation data set. So a couple of years ago, I read this paper that maybe some of you have read, or if you didn't, you should. It's called AI Can Be Sexist and Racist, Time to Make It Fair. So it's an interesting and kind of, punchline there title, but made me think a lot. So this was published in 2018, and it was basically talking about the biases that we, or nowadays, 2023, we know about these things. At the time, or even before that, we were not really conscious about that. This is an example that comes from NLP, like text, right? Nothing to do with images. But it shows this kind of bias. So for those that speak Spanish, here it says, ella es una investigadora, también escribe libros. Elle est un chercheur, elle a aussi écrit des livres, quelque chose comme ça. And when we translate that to English, and this is a screenshot from my phone, it was saying she's a researcher, he also writes books. And this is exactly what happened in 2018 when I read this paper. Because I took this screenshot. So what is it doing? Well, it's perpetrating certain biases that are there in our society, right? Whenever we talk about someone that is writing, it has to be a he. It shouldn't, but it is what the model was doing. Also in 2018, there was this paper, really important in the computer vision community, published by Timid Gebru, where they showed that for different facial recognition systems, if you look at the if you look at the performance of the model from an intersectional point of view, in the sense that you analyze how well the model, what is the error rate for the model in dark female, dark male, light female, and light male population, what you will see is that for dark females, the error rate for these commercial systems from IBM, Microsoft, what about 20%, 34%, while for light male, it was almost zero in many of these cases. Okay, so that is really telling us there is something here. The model is biased in the sense that it's producing low quality results for a particular demographic. Now, when I read this paper, and I was doing all this analysis with respect to medical images, I was working with medical images, I say, okay, what can happen in our context is even more dangerous, because I will put a system in production that will tell with 90% accuracy whether a person has a lung nodule or not for males, but maybe not for females. Can that also happen in our context? So at the time we started a research that ended up in this paper that appeared in PNAS in 2020, where we showed nothing new, but we systematically showed it with like a lot of experiments that, for example, gender imbalance in medical imaging datasets could also produce this type of bias classifier. So it was basically just raising the alarm for everyone out there saying, hey, be careful with these things. We can produce models that are biased and will not be providing the same standard of care to all the population. We then published this paper in Nature Comms, where we call the community basically to start thinking about that. That was great. So in our community, no one, not even ourselves, we're evaluating our models considering this kind of intersectional perspective, like saying at least, hey, does the model work as well in men and women, or in young people and old people? In the COVID era, we saw a lot of that. So there were these papers that were claiming that we are solving the pneumonia classification problem. And what they were solving was detecting the age of the patient, basically. Because all the pneumonia patients were older people, and then they were also testing on population where there are a lot of pediatric patients. And of course, the pediatric patients didn't have pneumonia, so the model was basically just telling us the age of the person instead of whether it has pneumonia or not. You know, these models are really good at capturing these shortcuts when they are learning. So there have been after that a lot of different works on this spectrum, but particularly in 2021 there was this work that was showing that segmentation models, like the ones I told you before, can also be biased. And in this paper, they showed that, for example, if you take heart segmentation in MR images and you consider different ethnicities of the patients, this is data coming from the UK Biobank, what you can see is that for white patients, the model was having a performance of about 93% dyes. But then when you go to Chinese, Black, other populations, the model was going much lower in performance. And this is really not something obvious. Why a model? I mean, there is no real difference in terms of the images between a Chinese, Black, or white person when it comes to the size or the shape of the heart, right? So we still don't really know why that was happening. I mean, this was happening because of underrepresentation of all these other races in the data set, so it was really correlated with that. But it's not clear why. I mean, these models, it was shown in another paper then, can predict race from X-ray images with a 95% accuracy. How can you predict a race from an X-ray image with such an accuracy? I really don't know why that is happening or that is possible, but it was shown it's possible, and if it's possible, most likely the method will use that as a shortcut to basically now say, hey, if most of the Black people have a certain disease, then I'm going to predict that just because it's Black, not because the certain disease is there. So we have to be really careful when we create these models. We also observed in our models, our own segmentation models, that they were pretty biased with respect to the age, for example. So here you see age, here you see DICE score, and what you can see is for really young and older people, the DICE score tends to be lower. And in this area here, it tends to be higher, okay? mostly because of an underrepresentation of pediatric and really old patients in our data. Now the point is, if you want to audit fairness, how you do that? How you audit fairness on a given segmentation model in this case, but it could be the same in other type of model. Well, you take your model, I mean it's really simple, and you evaluate the performance on the different subgroups that you are auditing for. Okay, so I take my segmentation model, I take a bunch of male patients, female patients, and I compute some kind of metric from that. For example, the gap in terms of that score. The DICE score, it measures the overlapping, right? Because I was telling at the beginning. So you can see that, for example, if there is a big gap between the DICE for this and this group, well, the method may be biased. The problem with that is that in order to compute this size, I need ground truth. So I need to have annotations, manual annotations in the new population where I'm deploying this system. Imagine I have a system that it's being used, approved by FDA, wherever, and it's being used in different hospitals. So I go to a new hospital, and if I want to audit the model in this new hospital to see if this is still fair, I will need manual annotations from doctors telling me the structure. So the question we tried to answer in this following up paper was, can we anticipate bias for segmentation in new populations without ground truth? Can we do that? And we propose a method based on what is called reverse classification accuracy. So reverse classification accuracy is a framework that was proposed a few years ago by Ben Glocker and his team. That is also based on simple ideas. So let's say I have this image, and I have this segmentation here. This is a predicted segmentation for some model. I want to know what's the quality of that segmentation. How can I do that without ground truth? Well, one could use like uncertain other type of approaches, okay, which are not the ones taken here. Here the idea is that you are going to have a different database, a previous database, for which you had annotations. So these are ground truth annotations. These are manual annotations. I know they are correct. So I have this reference database, and I have my image and the prediction I want to test. What you are going to do now is you are going to take this image, I, and you are going to register to all the images here. So in a deformable way, right? I take the image and I register with every one of those. I deform it so that it looks like that. And then I take this deformation field, the one I predicted to deform the image, and I apply to the images these segmentation maps here. So I bring this segmentation to the space of these other images for which I knew the quality, like the ground truth segmentation map. And what you do is to simply compute the DICE score, the overlapping or some metric between your propagated labels from that image and the ones that you have in your data. And then you take the maximum or the mean, you basically aggregate this metric somehow. This metric will be a really good, or in anatomical segmentation problems, will have high correlation with the real DICE. Why? The hypothesis here is that if the segmentation is good, it should be useful at least to segment some images from my data, for which I know the ground truth. So I can use that as an estimator of the segmentation quality. And if I can use that as an estimation of the segmentation quality, I can also use that to measure gaps in segmentation quality between different subgroups. And this is what we demonstrated in this paper. I'm not going to go into the details of the experiments we did, but we basically did like a strong validation to see that whatever gaps you find with real ground truth, you can recover these gaps with the ground truth, without ground truth and using this estimate. It has an enormous correlation, and basically it allows you to detect the same kind of biases that you were seeing when analyzing the behavior of the model of female or male separately. You can kind of recover the same tendency using this method. Again, I'm not going to go in the details because I think it's been too long already, but the method can be used for that. And interestingly, we use this method for auditing the bias in this huge data set that I was telling you that we segmented. So here, something we discovered, for example, that we didn't know. Here you are seeing at the distribution of DICE scores for different subpopulations in these data. And one of the subpopulations we considered was whether the image was taken posterior anterior or anterior posterior. So, right, when you an X-ray image, you know, you can take it with the machine here or with the machine here. And we observed this, that is, the dies for anterior or posterior, the distribution of estimated dies is lower than the distribution estimated for the posterior anterior. And why that happened? Well, when we told that to the medical doctors, they told us this made complete sense because anterior posterior images tend to come from hospitalized patients. You cannot move the hospitalized patient. You have to take the image from here. And so hospitalized patients usually have a lot of artifacts, tables, stuff like that, because they are in the hospital. And these artifacts are really make the task of segmentation much more challenging. So this is basically what our model was showing. There is a higher bias that we could discover thanks to this method, because otherwise we didn't have segmentation quality indices. We estimated it using that. So if you are using this group, you have to be careful because here the quality of the segmentations we are providing is much lower. Well, with that I conclude basically the talk. Simply mention other since I'm going to be here for a couple of more months, and maybe some of you work on areas that are interest. I only talked about this part, but we also work on this. So, for example, domain adaptation and generalization. Okay, when you want to basically, how can we obtain models that generalize well to unseen domains in images in this case? So there we have some publications on ITTV on different venues. Model calibration, this is another important aspect that I didn't cover. That is basically how can we get calibrated posteriors from our model? In the sense that, these models produce values between 0 and 1. But can we really trust these values between 0 and 1 to really be a probability of having a certain disease? Well, usually the models that we produce have really bad calibration, in the sense that the accuracy does not really correlate with the value that we are outputting there. So how can we improve that in the context of segmentation? Fairness, I already talked about that. And then we also work with other type of data, like image plans of images. This is a collaboration I'm having here with the Institute of Plant Sciences in Paris-Saclay, where we are basically trying to segment plant images to extract phenotypes from these images. This is, for example, a plant that is growing here. And you want to know the speed at which the plant is growing. You want to have basically a graph representing the structure of the plant. So this is basically, again, extracting graphs from images, and this is the methodological problem we are tackling. With this, for example, we could show, thanks to this system we devised, that plants grow faster during day than night. This is something that the biologists kind of knew, but they weren't able to prove it. And using this high throughput phenotyping method that we implemented, we could show that plants are growing faster during day when there is light than during night. And well, also some other projects where we try to relate these embeddings that I was telling you, shape embeddings, with genetic information. Basically trying to understand how certain changes in our, certain mutations in our DNA may impact the shape of your heart, in this case. Okay. So, yeah, this is all. These are our funders, and thank you very much.
Talk, and we are ready now for questions. Yes, please.
Yeah. Thank you very much. It was very interesting. For your first method, when you have your encoder in a segmentation network, do you have to take different levels for training both models, or you take
It's the same. You could use them. If you have a lot of annotations, for example, in a different image modality, let's say you are segmenting, I don't know, some structure in MR, but you have the annotations also in a different datasets in CT. Well, you can use these annotations because this only requires the annotation. It doesn't require the images themselves. So you could use the other annotations, pre-train your model with that, and then just continue. But otherwise, no, all what we did, it was with the same data. So I'm not sure how many people here works with medical images. I guess not many. So sorry for boring you. But I think this fairness part, it's really applicable to any work we are doing. I don't know in what domains you are working, but if you are doing NLP or if you are doing genetics or if you are doing whatever application, I think it's a really important topic to look at. So I hope that at least, yeah.
I have a question.
I also.
Okay, it's very good.
But you work on medical.
Yes. My question was is about this extra laws you add that enforces plausibility of anatomical structure. And my question would be like, is there any trade-off between weighting one or the other, or do you get some? Do you lose some kind of performance?
No, actually, you get performance at least in the experiments we did, because you are actually looking for shapes that are anatomically plausible. The problem is the model sometimes cannot really handle that because of the way it's trained. But of course, there is a balance between the two terms. If you only have the anatomically plausible term, you're going to get results that make sense globally, but not in the details, right? So you have to really find the sweet spot for this lambda by cross-validation or something like that.
But when you encode the image and then decode it as a graph,
Yeah.
so you give this example where you have two, so you learn separately the two of these, and then you kind of merge them together. But I'm not sure I get why you're not training directly the animal.
You have the point there. So actually, what I told is more kind of didactic. I think we started in that way. That's the way we conceive the model. But then we also start actually doing it from scratch. So you mean here, doing this from scratch directly. The only thing that doesn't have, I mean, you have better convergence, if you like faster convergence, if you initialize with the pre-trained model. So if you pre-train this, then you pre-train that, then you cut it. Of course, this latent space is not going to be aligned with this one because they were not trained at the same time. But if you connect them, because they have the same shape, and then you continue training, this model is going to converge faster, but not necessarily to better results than the model from training from scratch. But at the end of the day, we always train from scratch now, because this, even if it converts faster, you have another step of training here. So we should do it. But yeah, it's a really good point.
I have also one question. I like a lot of Ferrante's work, and I can really find applications on medical or on AI that been able to actually address this kind of biases that we can understand. For example, in the application with the plants, is there any meanings of measuring fairness on the
I mean, in general, when we talk about fairness, we are talking about human populations. I mean, we are talking about demographic attributes that define these groups, which, I mean, at least to me, yes. Otherwise, you are in a domain shift problem. Like, you are having just a domain shift problem, but I cannot really say it's unfair. Well, it's unfair with whom? Yeah, maybe with certain institute that is providing me the plants and not with the other one. But saying it's unfair with one plant or the other is a bit... Yeah, totally. I mean, you will see the same type of biases. I'm not sure if I will describe it as an unfair model, but just a biased model. To me, the fairness parts come into play when we are talking about humans.
Okay.
But this is to my conception of the problem. I don't know, maybe not. But yeah.
Any other question, even from the people online? I don't know if something.
No, so I cannot see the. Oh, okay. I turned off the computer. If they had any questions. No, but I think no. They are just putting in the chat.
Okay.
But no question.
So thank you very much.
Damian has one. Wait. Damian has one question. Can you use invert fairness and identify populations? Okay, Damian, thanks for the question. So You mean, I guess what you are meaning is what we call unsupervised bias discovery. That is basically discovering the clusters of, discovering the clusters of where the model is failing. Because here we know the protected attribute that we are using to define these intersectional groups. We are saying sex, or we are saying ethnicity, or we are saying age. But it may be also possible that you don't know these attributes for the population in which you are testing. So you may want to discover what are the failure modes of the model, basically, in which clusters the model is failing. Yes, you can do that. This is actually something on what we are working now. You can do that in different ways. You can use uncertainty, for example, to kind of say, okay, I don't have ground truth, I don't have this population, if you have ground truth, it's even easier. But if you don't have ground truth, you can use uncertainty to see what are the groups and then try to characterize the groups. How to characterize the groups is not something obvious. Let's say I have a bunch of images for which the model is failing. What do these images have in common that are making the model fail? It may be some characteristic of the population. With Maria now we are trying to use large language, like vision language models to do that. So this is like a new project we are starting with some students from here to basically describe in text what do these images have in common that are making this model fail. And this is like a new strategy that is called bias to text that has been starting to be used, using text to describe that. But I would say it's a really open question, really good one, and we are working on that.
I'm just going to open the mic for...
Yeah. There is a way in which we can open the mic? Okay, sorry.
There are questions in the chat.
Yeah, there. I think Mohsen had a question as well.
Yeah.
It doesn't allow to open the mic. But maybe...
Merci.
There it is. Reactivate. So I think you can unmute yourself, Mohsen.
Okay, thank you. Do you have my voice?
Yes, we hear you.
Okay, thank you for your great presentation, Mr. Ferrante. I saw that in hybrid GNet architecture, you use the Chebyshev spectral convolution. So for that case, that the hybrid class that you used and also your reference in GitHub, the number of samples is few. You talked about the big data. So considering the application of this method on big data, the question is if it works in big data or not. So we need some reference of computational cost, time computation, and the memory occupation of this method, as you explained in the hybrid method, is compared with the other methods. So it will give us idea to if we could use it in the ultra scale large data or not.
You mean in terms of memory consumption from the model when compared, for example, with the standard CNN?
Yes, I'm concerned. My concern is if I use it because it calculates the Chebyshev spectral convolution. It takes memory and time to calculate it. So I'm a little concerned of that.
Yeah, no, but this you compute it only once because basically these graphs, the Laplacian is constant, okay? Because we are defining the Laplacian of the graph just for the adjacent symmetric. So the adjacent symmetric is only one. Then what changes is the function that you define on the nodes. So this will be only computed once, and then you will always do the same. So actually, it's much cheaper using the model based on graphs than an NN unit, let's say.
Did you do the normalization of Laplacian by the random walk or other method?
We used the implementation that was in PyTorch geometric, so you can check there actually how it's done. We didn't implement it ourselves.
Okay. Thank you.
Welcome.